we're going to be talking about mutations, right? So DNA mutations. All right. So anyone know, like, what does the word mutation mean in general, right? So let's go to Anas. Right? So Anas, so what does the word mutation mean?
Change in appearance.
Yeah, some kind of change, right? If someone has mutated, if something has mutated, there's some kind of change in whatever that thing is, right? Right.
Wait,
So
is change in appearance like a official definition, or
not
is it
not
change in like the genotype or phenotype is my
so in the
question.
genotype. So
In the genotype, right?
right. So not necessarily in terms of appearance, but like I'm talking about in general, mutation means a change, right?
Change, right?
So DNA mutations or genetic mutations means some kind of change in the genetic material, in the DNA, in the chromosome, somewhere there, there's some kind of problem, right? There's some kind of change,
Uh,
right? So there are, so this is basically what a mutation is, right? So when a mutation occurs, what that means is that in the DNA, right, there's some kind of a mistake. There's some kind of a mistake in the DNA, and there's different types that could happen. You could have, just like one, tiny, change somewhere, right? You could have like, small amount of changes, like maybe a couple of bases, got, they put, the wrong one by mistake, something like that. Or maybe like huge stretches of the DNA, like maybe like an entire chromosome got doubled or got deleted, something like that, right? So anywhere from small mutations to large mutations, right? And so mutations occur for all kinds of different reasons. So they can occur just kind of randomly, spontaneous mutations, or if people are exposed to certain environmental effects, like let's suppose someone is works as a someone is giving a lot of X-rays, like in the dental office or in a hospital or something like that, right? If that person is always giving people X-rays, then over time that builds up, right? And so
yeah.
X-rays, they can go through the body and they can hit the DNA and they can damage it and cause something, something to change into another, right? You have things like UV light, which is like from sunlight, right? So people get skin cancer if they have too much sunlight. Different kinds of chemicals, the foods people eat, all different kinds of things can cause genetic mutations. So the first thing we're going to do is look at kind of like big mutations or like large mutations. This is in the entire chromosome, right? So if we have a chromosome, right, you can have large changes that occur within that chromosome. All right, so Yusuf, take a look at this one. The first one is chromosome deletion. All right. So what do you think happens here? Take a look at these pictures. What do you think a chromosome deletion is?
Um, it like loses one or some part. I mean, one part or like the whole of a chromosome.
Right. So take a look at the second picture. Can you describe, like, what do you see there?
Uh, so like that chromosome is breaking apart, and the genetic material in between is being like deleted. It's just being destroyed.
Right. So basically, like some part of the chromosome basically just gets deleted. Right. And so now that person, all those genes that were there, right, all the proteins that could have been made from that part of the chromosome, now those won't be made.
I have a question though, brother.
Yeah.
This can occur even after the person is born, or this only occurs before the person is born? This is like, are we talking about like during
Right.
the
Yeah. So
when the child is inside like the mother's womb, for example? Is that only then, or
no. So anytime it could happen. Yeah.
okay?
Yeah. So this could happen like almost any time. Right. So, all right. Let's go to the next one. All right. So Freya, what does the next one mean? Right. So it says chromosomal duplication. All right. So can you describe for us? Take a look at these pictures. What can you describe for us? What happened here?
Like a part of the thing is being duplicated, another one of it.
Right. So basically, maybe there's some stretch, right? If you take a look here, there's some stretch of the DNA, right? There's some stretch of the DNA, right? Maybe it could be small or it could be big. Maybe it has just like one gene, or maybe it has like a lot of genes in there, right? So whatever it is, that basically gets copied, and now all of a sudden you have two of those copies, right?
That's why some people have an extra chromosome, right?
So that's something different. So we can talk about that a little bit later.
All right.
So that's so this is basically within the chromosome, right? So it's not that now that we have an extra chromosome, that we have two
But
chromosomes.
doesn't it say, look, it says produces an extra copy of all or part of the chromosome.
Right, but so when this duplicates, you'll have a duplicate within this chromosome. So maybe everything gets doubled, but it's still all
But it's
attached together.
Only all. Oh, okay, I see what you're saying.
Right? So you still have one piece. It could be a bigger piece, right?
Yeah.
It could be a bigger, chromosome now because now you have extra copies of things. However, it's still one chromosome, right? All right. So here we have the next one, chromosomal inversion. All right. So Anas. All right. So this one's going to be yours. All right. So what is a chromosomal inversion? What does that mean?
Inversion means, what do you call it? Like it reverses?
Kind of. So take a look at this second picture, right? So what's going on in the second picture? What are they showing happening in the second picture?
The chromosome is turning around and it's connecting the places like different.
Right. So what does inversion mean? Just as a general term, inversion. What do you think that means?
Opposite of something.
Have you heard the word
What do you call it? Yeah, invert, like opposite.
Yeah, kind of the opposite, right? Basically something that's like turned inside out. Like, have you heard of like an inverted glove or something? Like, this was like right when,
Yes.
when you take off a glove, it like turns inside out, right? So
Yeah.
that's kind of what inversion means. It basically means like upside down, inside out, something like that, right? So if you take a look at the second picture, right, what happened? Part of the chromosome like flipped, right?
Yes.
Part of the chromosome flipped, right?
Like the ABC turned into ACB.
Right. You see how B, so the rest of the chromosome is fine, but B and C, they kind of like turn upside down, and now they're going in the opposite order, right? So this picture on this picture over here on the left, this is kind of showing you like how it kind of happens. This is one model they have of how, this happens. So basically, these chromosomes, these two chromosomes, which are homologous chromosomes, they basically, kind of like come together and somehow one part joins to another part, and then somehow it cuts and flips into the other one. So there's different ways that maybe this could happen, but they're just kind of showing you how somehow, different parts of the homologous chromosomes, they get attached, and then, one part kind of cuts the other, and then they flip. Okay. This is showing you here, this second picture is showing you like a little bit more detail, or it's showing you two different types. One is called paracentric inversion, and one is called pericentric inversion, right? So basically kind of in the middle of the chromosome, not necessarily in the middle in terms of like distance, but basically in the middle of the chromosome is called the centromere.
Centromere.
This is where this chromosome can kind of join to the other homologous chromosome, right? So this is basically where these two can join together, and when it when you have the sister chromatids. All right, so Afnan, where's Afnan? Right,
You left.
right. So
Wait. Anas, are you at ISOM right now?
No.
I don't think anybody was here today.
Really?
Yeah. So in this one, paracentric, right? The part that flips, it's not here at the central centromere. In the pericentric, right, it flips right around the centromere, right? So I don't think you have
Right
to,
on it?
yeah,
Okay, right around
yeah,
it.
right. So that's part of the segment that flips. That's what it means. So, so I don't think you have to know that, but that's just like those are two different types of inversions. All right, so we have translocation, right? So what does translocation mean? Translocation kind of sounds like transportation, right? It's like one thing moves somewhere else. So a part of one chromosome breaks off and goes somewhere else, right? So for example, over here, right, you have, this original chromosome A, B, C, D, E, F, right? And then this small piece from somewhere else, GH, shows up and just inserts itself right here in this chromosome. All right? So this is basically, this GH, right, isn't really supposed to be here in this chromosome. It came from somewhere else, but now it got translocated into this chromosome. All right? So there's different kinds of translocations. This picture on the bottom right, that's giving you one example. All right? So this is called a reciprocal translocation. Right? Fariha, what does the word reciprocal mean, or like reciprocity, or something like that? You see these kinds of similar words? What do these kind of words mean?
Opposite or like turning around.
Opposite, right? Yusuf, what do you think? Where have you heard the word like reciprocal or reciprocity?
Like in math, but like it's like this an inverse thing, but it basically means the same thing, but it's just inversed or something like that, I guess.
Yeah. So basically, in general, reciprocity means that, like, it's usually like a transaction. Like if you give someone something, they give you something, right? That's called reciprocity. So
because like they're both trading parts
Yeah.
with each other.
right. So if you go to the store, right, the store gives you something, right? The store gives you like an item, and then you give them money, right? So it's like each person giving the other. So this is called reciprocal translocation. You see the green one and the yellow one gives the other,
Trading.
a part of the chromosome and then takes from the other, right? So this is like one type of translocation, just an example. Oh yeah, we forgot to do. Oh yeah, so we'll do the quiz from last time at the end. Right? I forgot to do the quiz. All right, so gene mutations. Right. So now what we talked about before, right? What we just talked about. This was like on a big scale, right? This was on like a large scale, right? Like entire, entire pieces of chromosomes are being, moved, flipped, stuff like that, right? So that's talking on a large scale, right?
But brother, aren't genes inside the chromosomes?
Right, but so that's still on a kind of like a larger scale. That's like a gene is like made up of usually like hundreds or thousands of base pairs. Like A, T, G, C, G, like thousands of those make like a gene.
But a chromosome is made of like hundreds of thousands of genes,
of
right?
genes, right?
Or
So-
like thousands, or
yeah,
just 100.
Yeah, usually like thousands of genes on a chromosome, right?
So then, how are gene mutations even worse?
Right. But first of all, I'm just saying that. So that was like on a larger scale. Okay. Now we're going to go down.
Oh,
to
that was on a larger scale.
Yeah.
All
So
right.
now we're going down to the small scale. Like you see, we're talking about individual bases, right? A, T, G,
Yeah.
C, right? These are the individual bases. So now we're going to talk about mutations or changes down at this small level. All right. So let's take a look at this example, right? So let's suppose that we have this thing UUA, all right? Now, remember we talked about yesterday that when you have a messenger RNA, right? There's, every 3, bases codes for an amino acid, all right? So for example, this one right here, UUA, right? Let's suppose this messenger RNA is going through the ribosome, right? If it sees UUA, then this amino acid leucine is going to be attached in the protein, into the protein. All right. So let me go back. I don't remember where I put those. All right. So do you guys remember? Let me see what's coming up. All right, so on this screen is showing. You guys remember this table over here on the right?
Yes, brother.
yeah. The amino
Codon
acid,
chart.
the different base, the codon chart.
Right. So this right here on the right, this over here is telling you that, like for example, if you have CUU, right, you're going to get leucine or whatever, right? The different combinations are going to give you different amino acids, right?
Different amino acids.
Right? Now, so what happens if, let's suppose it's supposed to read, right? Let's take a look at this one. What if it's supposed to read CUU, right? So it's supposed to read CUU right here, right? So that's supposed to give you leucine, right? That amino acid leucine is supposed to be there, right?
Uh-huh.
But what if like a mistake is made, right? You see that middle U, right? What if instead of that middle U, you got a C, right? It's going to look like this one on the green, right?
Yeah.
Right? Instead of CUU, it's going to look like that one, CCU, right? This was just only that one mistake was made. The leucine is going to turn into this one, proline, right? You guys see that?
Yeah.
So this is what we're talking about, right? So there's different kinds of mutations that can occur. All right, so let's go back up here and take a look, right? So that this kind of mutation where we're talking about like one thing changing into another thing, like one base changing into another base, all right? This is called a point mutation because this is on a very individual level, right? So we're not talking about chromosomal mutations. These are very small point mutations, right? So these are some of the different examples, right? So let's take a look at the first one. So the first one, what happened here? All right, is it still connected over here? All right, is it still connected over here? Anas and Yusuf?
Yes.
Okay.
What's connected? Yeah, we're good.
Okay, I think the other computer got disconnected. All right. So let's take a look at this first one here. All right. So the first one is frame shift mutation. All right. So what do you think happened here? All right. So let's take a look. All right. So Freeha, we're on the first one. So the first one says frame shift mutation. All right. So Anas, so what do you think happened here? Like, what's the mutation? What's the change here?
Frame shift. They probably got added a letter.
Exactly, right? So you see this G got inserted in the middle, right? All right, so this one we have this one extra that got inserted in here, all right? How about this one over here, all right? What about these three over here, right? You see these three over here? All right, Yusuf, what happened in these three over here?
Substitution, brother.
Something got substituted, right? Instead of this first U, right? Maybe it changed into a C, right? Or it changed into a G. Or this middle U changed into an A,
Uh-huh.
right? So when that happens, like different kinds of things can happen, all right? So first of all, let's take a look at this picture over here, right? So in this case, right, we're looking at before the mutation and then after the mutation, right? So after the mutation, right, basically one thing got substituted for another, right? So there was supposed to be a C here, but all of a sudden a T took its place. And so what happens when we do that, right? You see the amino acid can change, right? Because the GTA codes for this thing, whereas GCA codes for a different amino acid,
Uh-huh.
right? Right? Now, this word codon, right? You see each of these three things, TAC,
That's a codon.
GCA, right? Each set of three that codes for an amino acid, that's called a codon, all right? All right. So there's different kinds of, so let's suppose we substitute one thing for another, all right? So different kinds of things can happen. The first one is called missense mutation, right? So let's take a look at this chain over here, all right? There's these are the amino acids, all right? So Freeha, can you tell me when there's a missense mutation from the first to the second, all right? What changed? Can you tell me, like in effect, like what exactly? All right, Freeha, what exactly changed from the first protein to the second protein?
A got substituted with
All right, so A got substituted with C, all right? Now, but what happened in the protein, right? So that's in the DNA or RNA, but what ends up happening in the protein?
another
Another what?
incorrect
Incorrect, which one?
amino acid. There's an incorrect protein.
Right, so do you see here that, when we're coding for it, right? These amino acids, right? Most of them stay the same except for only this one right here. Only the proline is different, right? So all of these are made of these histidines. These are amino acids. So most of them are the same in the bottom one, except for the one that changed. That became proline. Does that make sense?
Uh-huh.
All right. So that in missense mutation, only one thing changes. That's very important. All right. Only one thing changes. Right. Now usually there's some kind of problem that occurs if that happens. Right. Sometimes there's no problem. Right. Sometimes because a protein, when it forms a string, eventually it folds into a 3D shape. Right. So maybe this amino acid, even though it changes the shape a little bit, overall the protein can still function. So sometimes it produces a protein that doesn't function at all. Sometimes it produces a protein that functions a little bit less. Sometimes there's even no change. All right? So this is one kind of mutation, missense mutation. All right. All right. Let's go back up here. All right. So this one over here, you see this one in the middle, it says missense mutation. That's what we just talked about. Missense mutation. One amino acid changes into another. All right. How about this one over here? Silent How about this one over here?
Silent mutation and missense mutation are the same thing, right?
Right. So you take a look at it. It says silent mutation, right? So from the first one, the U changed to a C, but basically there's so even though this changes, right, this causes no problems at all. Why do you think that's the case, right? Anas, why do you think that's the case, right? Even though there's this change, right, this U changed to a C, but there's not going to be any change in the effect. Why is that the case?
Because it probably gives up the same protein or whatever.
Mm-hmm. All right, so let's take a look at this one, right? So what do you notice about this table? Take a look at this table here. Take a look at this table here. Right? So can you explain using the table?
Can you go back to the question?
Mm-hmm.
Oh, brother.
Right, so take a look.
From UUA to CUA.
Right, so
Can you go back to the codon chart?
So UUA to CUA.
So U and because they both produce the same allele.
Exactly. Right. Right. So the same amino acid. You see how each amino acid is coded by multiple things. It can be coded by multiple codes, right?
But then it's not a mutation, is it?
So basically, if you put UUA, you're going to get leucine. If you change that to CUA, you're still going to get leucine.
Right? But it's not a mutation, right?
So it's still a genetic mutation, but it's called a silent mutation. That's why it's called silent mutation, right?
It doesn't affect it.
It doesn't affect it,
right?
right? Does that make sense, right? So that's called a silent mutation, right? Now, let's take a look at this one up here. The last one, all right? So take a look at the last one. What does that mean? Nonsense mutation. This is nonsense mutation. Stop codon. What does that mean?
This is like a what's it called?
So UUA
thing that doesn't
change-
exist.
So let's go to UUA goes to UAA. All right, so let's go back to let's go back to this picture here. Where's UUA?
You mean
No, sorry,
UAA?
yeah, UAA. Where's UAA?
So we go in UA.
Wait, I don't know if
Yeah, there's a stop codon thing there.
Yeah, so UAA.
Oh, now I get it.
So So what do you think stop codon means, right? So if it changes to UAA from UUA to UAA, this becomes a stop codon. What does that mean, stop codon?
It's like
It stops making
it's like the promoter. It's like the
amino
promoter.
acids.
The protein.
It's the same thing, right?
Exactly, right? So the amino acids are being formed. So if you get a stop codon, that's a signal that, hey, this is the end of the protein, stop. So, usually when that happens, that's appropriate, right? It's supposed to stop there because you don't want to keep going forever. So it knows where to stop. But sometimes that occurs earlier on, and all of a sudden you get like a really short protein. And obviously that usually doesn't work or function very well. So this is called this thing is called nonsense mutation. The protein just becomes nonsense, something like that. Right? Now, in these ones, in these three that we've looked at, right, when one thing is replaced with another, right, usually just like one, basically it's like one amino acid changes, usually, right, unless it's a nonsense, right, but usually like one thing can change into another. But what happens?
Each one of these is amino acids.
Right, but what happens to all of the amino acids afterwards? Do they change or do they stay the same?
What?
Huh?
What about all the amino acids afterwards?
What do you mean?
Right, so let's suppose we substitute this one over here, right? Let's
Oh.
look here, and I substitute one thing for another, all right? Look at the amino
Right.
acids afterwards. Are they going to change or are they going to be the same?
They're gonna change, man. You substituted one amino acid.
Right, no, so I'm saying this one amino acid changes, but what about all the ones afterwards?
No, I don't think so.
No,
No.
they're going to stay the same, right? So this one, this arginine, is going to change to the histidine, into the yellow. But you see the ones afterwards, the two afterwards, and whatever afterwards, there's no mutation there, right? So those are going to stay
Yeah.
the same, right? Now, there's another kind of
Yes.
mutation, right? Remember the first one we talked about, insertion, right? Where we
Yeah,
actually
frame
insert
shift.
one in the middle. That's called a
Yeah.
frame shift mutation. So that one can actually be
We change the whole thing afterwards
a
too.
lot more
Then
dangerous, right? So Anas, explain to us what happens. Like, how come this changes like everything?
because it adds another. What do you call it? It'll make a whole new code.
Base pair. Adds a whole new base pair.
Exactly, right? So take a look at this one right here, right? So they inserted an A, right? An extra A got inserted, right? Now remember, every 3 codons gives you an amino acid, right? But in this case, you see how the whole code shifted over? So every codon afterwards is now different, right? Look at the first one, right? So we put an A right here in the beginning, right? So originally, this codon was GCA. GCA. But if I add the A here, the GCA gets shifted over. And take a look at the codons. You see these groups of 3? Now you see that if you shift everything in the codons, now all the amino acids afterwards are pretty much all going to be different. Right? so I think we're going to be back. All right. Is it working now? Can you guys hear me?
Hello, brother. Yeah, I am the host now.
Okay. All right. All right. So I think the.
I'll pass. I will pass on the duty of being a host to you. All right, brother.
All right, Sean.
I'm kidding, brother. I just withdrew your co-host permissions.
Okay. All right.
Okay, I'll make you host here.
Uh,
All right. So I just put the reclaim host. All right. All right. So you guys understand? All right. So just as a recap, right? So if you insert one here, right? You see how everything shifts over, right? So every group of three is a codon, right? So usually the three, every three, so basically what happens is when you add an extra one, everything shifts over, right? So now you have, entirely new sets of codons, right?
yeah.
Right? Freehead, do you understand what happened here? Like why everything changes, even if you insert one thing here, everything afterwards changes. Does that make sense? All right? So this is insertion or deletion, all right? All right. So this is kind of showing you the same thing, right? If you have, if there's just like one thing that changes, right? If one thing changes into another, then it's just one amino acid changes. But if you insert one or take one out, all of a sudden everything afterwards changes as well. Right? All right, and this is basically kind of showing you, this is kind of a summary of the different kinds of mutations. So in the beginning is the normal one, then you have substitution, only one thing changes. But if you have an insertion or a deletion, right, then basically that thing changes, but then everything afterwards changes as well. All right. So there's different kinds of mutations can occur in different places. All right. So you can have what are called somatic mutations or germline mutations. All right. So a somatic mutation is basically in just some random set of cells. Could be the skin cells, could be heart cells, liver cells, could be anything. All right. Now in the germline mutation, what happens is that in the cells that are used to create the new organism, if there's a mutation there, then the baby that's formed, right, or the child that's formed, the baby animal or whatever, now that entire animal is going to have that mutation.
Okay, I'm confused. Germline mutations occur in the reproductive organs, or is it like
right in the germ cells,
in the whole body?
Right, in the gametes. So
it occurs in the gametes.
Right. So for example, let's suppose it occurs in, when meiosis occurs, right? Let's suppose a mutation occurs there, right? So if a mutation occurs there, then basically, the baby organism that's formed from those germ cells, are going to entirely have that mutation, right? But let's suppose in this embryo, there's some mutation that occurs in this embryo in this small portion. Well, then only a portion of that person is going to have that problem, but this person's children are not going to have that mutation, are not going to have that issue. All right. So let's we're going to look at like different kinds of mutations and stuff like that. So there's this idea called gene expression, right? So let me ask you a question, Yusuf, right? Let's suppose we have a liver cell, and let's suppose we have a heart cell. All right. So let's suppose, does a person's liver cell have a different set of DNA than the DNA in a person's heart cell?
Yeah, I guess, yeah.
Yes, all right. And why do you think so?
Different part of the DNA is applied to that part. But if you're talking it has 23 different chromosomes and a whole different DNA, then no.
Right. So the DNA is exactly the same, right? If there's the 23 sets of chromosomes, is the same in every cell, basically, right? except for doing
Wait,
meiosis
every
and
cell
whatever.
has 23 chromosomes?
Right, 23 pairs of chromosomes, right? So every
Then
cell
how does the...
so every cell
Oh,
has
because they're
the
the same exact chromosomes. Okay.
exact same chromosomes in every cell, liver cell versus heart cell. But so how is it that one cell becomes a liver cell and one cell becomes a heart cell, right? Doesn't a liver cell need different kinds of proteins than a heart cell?
Yeah.
So how do we reconcile that?
I guess the part of the DNA that's taken for a liver cell is different from the part that of the DNA that's taken for a heart cell.
Right, Anas, what do you think? So how is it that, don't they need, like, if these cells need different proteins and different things, then how do we reconcile that with the fact that the DNA in both is exactly the same?
The RNA code.
RNA code, so something to do with that, right? So Freeha, what do you think? Sorry? So I'm asking, so in like liver cell and heart cell, the DNA is exactly the same, right? So how is that the case if a liver cell, needs different kinds of proteins and things than a heart cell? So the DNA is exactly the same, but does that, so some of you guys, I think you guys, the structure is different, so something like that. So I think some of you guys got it, got some of the idea, right? So the DNA is exactly the same, right? But does that mean that in every cell, all the DNA is being used all the time?
No.
No, right? So there's like a whole bunch
Exactly,
of
part of the
proteins in
DNA.
the DNA, right? But but you pick and choose what kinds of proteins you want to use in this cell. So the DNA only creates certain RNAs and certain proteins, and the rest, it just stays there packaged up and is not used. So every cell has the exact same DNA. They have the proteins for any cell. However, the heart cell is going to express only certain genes, right? Gene expression, the term gene expression is going to express only certain genes. And then maybe like a liver cell is going to express different sets of genes. So that's what this idea of gene expression is, right? So you see this picture down here at the bottom, right? So from DNA to the protein and all these things, there's like a couple different steps in between, right? Right? It's not just the DNA itself does all the work, right? The DNA turns into RNA, which turns into protein. So there's regulation at each of these steps. The cell decides, hey, we're going to allow this to go forward, or no, we're going to stop this process and do something else, right? So the first step is obviously the DNA. The cell, the liver cell is going to decide, hey, we're going to open up this piece of DNA and make this protein. Whereas in a heart cell, probably a lot of those are closed up and not used, and it's going to open up and use other parts of the DNA.
So basically, each cell can decide what part of the DNA to use, right?
Right. Each
Or
cell
different parts of the DNA.
Correct. Each cell decides what part to use. So that's called gene expression, right? Not all the genes are expressed in all the cells. Some genes are expressed in some cells, and some genes are expressed in other cells. Right? So, when we talk about mutations, we've been talking about mutations, right? So, generally when you think of mutations, you think of like harmful mutations, right? So, for example, some of you guys may have heard of different diseases, cystic fibrosis and sickle cell anemia. Anyone heard of these kinds of diseases? Anyone know what these diseases are?
Yeah, I've heard some of these.
Some of these, right? What do you know about these cystic fibrosis or sickle cell anemia? What have you heard about these?
Cystic fibrosis, I think I heard it was related to the bones, but I'm not sure.
Bones, right? How about sickle cell anemia? Anas, have you heard of anything like this? You're not sure?
No, brother.
Not sure. So these are, so we'll talk about these diseases a little bit later. So these diseases, right? These are, usually when something changes in a protein, usually most of the time it's like something harmful, right? Now, there's some kinds of beneficial mutations as well. So beneficial mutation can change something to make it a little bit better. So bacteria, they often have a lot of good mutations, right? So, for example, something changes in them such that they can resist the effects of antibiotics a little bit better, right? So most bacteria are killed by an antibiotic, but some of them they randomly change. And some of those random changes can help them to survive some kinds of antibiotics. So, so sometimes when you use antibiotics, eventually over a few decades, maybe 20, 30 years, the antibiotic will no longer be useful because the bacteria have changed. All right. So that's an example of like a mutation that's beneficial for that organism. Right. So let's look at some like a few genetic diseases. Right. This is called sickle cell anemia. Right. So take a look at these pictures. Right. So you see the normal blood cells, and then you see these, sickled cells. Right. You see these ones that are like kind of long and thin. The structure inside has kind of changed. Right?
Oh, they're like little sickles. All right, I see.
Right. So as they're flowing through, they don't just flow through nicely through the blood vessels. You see in this picture over here, sometimes, these sickle cells, their abnormal shape can cause them to become lodged in the blood vessels, and then it blocks the flow. It stops the flow, and you get clots and pain and stuff like that, right? So saying blocked blood flow can cause pain, serious infections, and organ damage, right? So if you block the blood flow to an organ, like the liver, all of a sudden that organ is going to become damaged, right? So people can have these things they're called sickle cell crises, and people all of a sudden they have like a lot of chest pain somewhere, muscles, whatever it is, and so this can cause a lot of problems. Now the interesting thing is that some people, even though they have these problems, if a person has sickle cell anemia, then it makes them more resistant to malaria, right? Freya, what is malaria? Have you heard of malaria before? Not sure. So what kind of animal, what kind of insect, transfers malaria? Do you know?
Mosquitoes.
So mosquitoes.
What you're asking?
So like a mosquito, a lot of mosquitoes in tropical areas around the world, they transfer lots of diseases, right, including malaria. So there's a parasite that causes malaria. So but if a person has this sickle cell anemia, then actually they can sometimes be more resistant to malaria. Okay?
Do you know why? I'm just interested.
I'm not sure why. Probably someone has researched it. Maybe that could be your science project. We'll do a science project. Study.
Inshallah.
Right?
If they tell me, I will probably do it.
Now this one here is called disjunction, right? So do you remember when we were talking about meiosis, right? So when we were talking about meiosis, right, what happens, right? There's 2 phases, and eventually you make 4 cells, right? Now basically the idea is that in meiosis, if you take a look here, right, basically each time you're basically splitting the number of chromosomes in half, splitting them in half, right? But take a look at what happens over here. Let's look at the second column. It says disjunction in meiosis 1. So Anas, what happened in this portion here? What's like the problem they occurred here?
Both of them connected instead of separating.
Exactly, right? So the spindle fibers were supposed to pull them apart, right? But instead, it got stuck, and then this one moved all the way to the left, and there's nothing on the right. So basically, like if you go downstream from there, the gametes that are going to form are going to have some extra chromosomes. You guys see that? Right?
So this happens. This is how people have extra chromosomes, right?
Right. This is how people could have extra chromosomes.
Extra chromosomes.
Right? Some people they have extra chromosomes. So this is how that could happen, right? If the gamete that's used had an extra chromosome, well then the child is going to have an extra chromosome. Instead of having 23 pairs, they're going to have 23 sets, but they're not all going to be pairs. One of them could have like 3 or 4 in there. Right? Right? There's usually supposed to be 2 of each, right? Now maybe some of them might have 3. Right? So this is basically what non-disjunction kind of means. And so this results in different kinds of syndromes. There's a lot of syndromes, for example, Down syndrome. This is the kind of thing that happened. I don't remember which number. I don't remember if it was number 10 or which number chromosome it's at.
21.
21. Oh yeah, that's right, 21. So then there's other ones as well. There's other syndromes where at different. So basically, if you double, if you have an extra chromosome at other chromosome numbers, you can have other syndromes. All right. So Down syndrome is like the most like well-known type of syndrome of this type.
Mm-hmm.
Right. So here we have, the next one is called cystic fibrosis. So if you look over here, in the bottom right, you see over here, right?
Mm-hmm.
Basically this entire codon CTT, right? You see this entire not entire codon, but these three right here, these three bases, they just basically can get deleted, right? So now cystic fibrosis could be caused by different kinds of mutations. There's not just one type that causes it. But this is one example, right? Basically these three in the red, they basically just get deleted, they're gone. And so basically the protein that's formed is like a slightly smaller, and sometimes it doesn't work very well. So people here in cystic fibrosis, in their lungs and in different places where like fluid is supposed to flow like nicely, it gets all clogged up, right? Because you can't get water into these areas very well, and so just instead of become watery, and you can quickly cough up mucus and stuff like that, it just gets stuck there, right? Because you can't get water into those places, right? So this is called cystic fibrosis.
And it is most common amongst Northern or Central European descent, but they're unsafe for them.
Right. Yeah. So usually among North European descent. So this protein is called CFTR. This is a protein that transports chloride, right? So this is all right. Now insects, right? Just like we talked about, bacteria can form mutations to resist against, antibiotics. Insects, same thing. They can form mutations, and all of a sudden you could have an insect that's better adapted to some insecticide. So a farmer might spray like an insecticide all over the plant, right? And then after several years might notice that, hey, the insects are not really dying off as much, and so eventually the insects come back, and then you have to use some other chemical, right? So same idea can happen in insects.
So like the insects are kind of immune to the thing.
Right. So in human beings, right, they're just mentioning that in different kinds of diseases, right, some people, could have, some mutation that causes them to be resistant to certain diseases, or have, increased, bone density and stuff like that. So different kinds of random mutations could, cause different kinds of, changes.
But there's probably like a downside to it too, or like there's no trade-off or anything like that.
Yeah, sometimes there's different downsides as well. Like for example, we talk about malaria versus sickle cell, right? So the downside is that you would have the sickle cell.
You have sickle cells, but you're safe from malaria, or like a little bit better.
Relatively,
Yeah.
Now, there's something called. So remember, in human beings and most animals are diploid, right? Anas, what does that mean, diploid? If I say that human beings are diploid, what does that mean?
They have more chromosomes?
You have, at every chromosome, you have how many chromosomes? Every chromosome number?
2.
2, right? So polyploid, right, means that you have like more than 2. So this one
like any amount more than 2.
Yeah, so this one has like a 6 at every chromosome. This has 6. So they're saying over here that sometimes in citrus fruits, like when people try to breed better plants, they found that this thing polyploidy can be beneficial in citrus plants. That it makes the plants a lot more resistant to problems and stuff like that, like stronger plants and stuff like that. So I guess in plants, this when they breed plants, it can make fruit that's better than before.
Oh.
Right. So now we talk about mutations, right? Like changes. So what causes mutations? Those things are called mutagens, right? So these are some examples of different things that can cause mutagens, right? So we talk about X-rays, UV light, right? So carcinogens, right? From cigarettes, like if like a lot of smoke, right? A lot of smoke, whether especially from cigarettes and other kinds of things that people smoke, that can cause cancer. They noticed that in processed meats, that there are certain kinds of chemicals called nitrates that maybe could cause like colon cancer and stomach cancer and stuff like that. Different kinds of cancers in the digestive system.
Oh.
Right. So in fact, so one thing, so it's better to eat food that's not processed, that is not packaged up and stuff like that, where they don't have added chemicals and stuff like that. and it's important. That's why they always say that whenever eating meat, always eat a lot of vegetables with the meat, right? So in fact, so
It depends on where you get your meat from, right? If you're not getting
Right.
processed meat, then you're good.
right, but even if you eat if you eat regular meat, it's still recommended to eat vegetables in the same meal.
Vegetables with that meat.
Right. So in the same meal, eat like meat and vegetables. So one of the best like vegetables to fight cancer, so there's some that are very good, like broccoli is very strong, stuff like that. Red onions are supposed to be like one of the onions, especially red onions, are supposed to be one of the best ones. So I don't know if it's like the strongest, but they found that red onions, they kill like a huge variety of cancers. At least in the laboratory and stuff like that. When studying it, it seems like the chemicals in there kill like a huge variety of different cancer cells, right?
But it still isn't a cure to cancer or anything like that, right? You can't cure cancer. I mean, people haven't really
not
found a
necessarily
cure.
But so this is more like for prevention, right? I'm talking about
Prevention.
when you have like low levels, right? Normally, so every person like normally gets like some mutations and stuff like that, but usually the body recognizes it and gets rid of it while it's small. While you have a few cells, the cells are acting weird, the body gets rid of them, right?
Yeah.
But so when it's at the low level, you want to eat a lot of fruits and vegetables to prevent it from becoming bigger.
From becoming
Right.
higher. Yeah. Okay.
So it's not necessarily a cure for cancer, but like prevention is probably more important.
prevention is important, right?
Right. So different kinds of things can
There's
cause.
something called chemotherapy for cancer, right?
Right. So that's because once you get to that point, then a
It's
lot
like
of
a
cancers.
very large scale cancer.
Yeah, a lot
It's like
of
large scale
cancers
mutation.
with the medicines, right? Even it's still like only like 50% or sometimes less survival rate.
Yeah.
Right.
Yeah, it's like very small
Right.
percent chance that you're gonna be fine.
Yeah. Right. So this is cancer, right? So we talked about cancer a little bit, right? So what happens in cancer, right? So in cancer, basically you have different kinds of mutations. Now most cancers you have multiple mutations, all right? So the mutations, they especially occur in basically what are called like tumor suppressor genes. So what are these genes? These are genes that control cell growth, right? They're not usually controlling some random protein. They're usually controlling cell growth. So Freeha, why do you think that's important to know? Why is it that cancer mutations usually occur in genes that are responsible for controlling cell growth? Freeha,
Wait.
why do you think that's important? Right. So basically, like, what is cancer? It's like cells are out of control in growth, right? They're just growing all over the place, right? So if you have a mutation in some random protein, maybe it doesn't do anything. But if you have a mutation,
But then, how is cancer any different from a tumor?
no. I mean, tumor is it's kind of like a tumor is kind of like a large collection of cells. That's the term.
But then, cancer is when you.
So cancer
the
is kind of like a
cells-
general term for this whole thing, like different kinds of tumors, malignant tumors, metastatic tumors, just kind of like a general term.
Okay.
Most usually physicians, they don't usually they usually use like more specific terms to talk about things in more detail. So
all right.
so this thing over here, so basically if you have mutations in the genes, the proteins that control.
Oh, Ibrahim is there, and you're here.
Yes.
Huh?
Yeah, right. Yeah.
Oh,
So usually if you have a problem in these genes, right, then what happens, these cells, right, there's nothing stopping them from growing out of control, right? Usually these genes are stopping and regulating how the cells are growing, right? So if it grows too much, then it stops it, right? Remember the cell cycle we talked about, right? So it's very regulated, right? The cell can't just keep going through and through forever and ever, right?
Yeah.
So, but if there's a problem there, then those stop signs are not, followed, and so the cells, they just keep growing, and you have a problem. Now, most of the time, it's not just one mutation in a cell that automatically leads to cancer. Usually, there's a whole set of mutations, right? So, for example, let's suppose there's some cell damage, right? You get the first mutation. So then, now this cell is growing, and but then in some of those cells, another mutation occurs.
Another mutation.
And so eventually, after a couple of mutations, then
You
there's
have too
nothing
many
stopping that
mutations.
cell from from not growing, and so it just starts to grow and becomes, some kind of cancerous kind of cell.
Oh.
Right. All right. So this is saying that in certain kinds of tumor suppressor genes, that if you have a problem, then cell division is not under control.
Oh. All right.
Right. So let me see what else. So there's one gene. They mentioned a couple of details here. So they mentioned here that viruses. So viruses, sometimes there's certain cancers that are associated with viruses. There's certain cancers associated with viruses because viruses, what they do. So we haven't. I don't think we've talked about this in the thing, but viruses, they actually inject their DNA into a human cell's DNA. So all of a
But
sudden,
aren't
if you're
viruses non-living?
what happened?
Viruses are non-living. They're not alive.
Right, but they're kind of like a machine. So the machine goes, attaches to the cell, and injects its DNA in. So obviously,
But
if you're
then non-living things don't have DNA, right?
yeah, so obviously, if that happens, then that new cell now has this new weird kind of DNA, and sometimes there's problems there.
Yeah.
Right. So virus infections can cause DNA, can cause cancer. Smoking can cause cancer. And then also, so there's a gene called P53 that they mention here. That's like a very famous gene. So a lot of cancers, they have a problem with this gene. Right? So this is one of those tumor suppressor genes. It's responsible for delaying the cell cycle until all chromosomes are replicated. So if that's kind of like a stop sign, like, hey, stop here for just a second. So the cell ignores that stop sign, just keeps going because that's mutated. Then that's like something that that cell is not controlled. It's getting out of control. All right. So I think that's it for today. So all right. So we'll have to